Good morning. We are Group 4 and today we are going to be discussing animal cell culture. In this report, we will be explaining cell cultures and its types. We will also describe the workings in a cell culture lab and describe the fundamental techniques such as reviving, passaging, and cryofreezing cells. So first, let's discuss the basics of cell culture. Cell culture refers to the process when cells are grown in controlled conditions outside of its native environment. That's why it's always considered an in vitro procedure. The culturing of cells has many applications, such as investigating disease models, testing drug candidates, and producing biotherapeutic products, to name a few. Nowadays, cell culture is widely used in laboratory settings, especially in vaccine research, cancer research, and protein therapeutics. Cells used in the labs can be classified into two: primary cells and established cell lines. Primary cells are directly obtained from an organism's tissues. When grown in the right conditions, primary cells can grow and proliferate, but can only do this a finite number of times. This is because each time they divide, part of its DNA telomere is lost. After a certain number of cell divisions, the cells eventually reach senescence, wherein the cells can no longer divide. On the other hand, established cell lines are obtained from clinical tumors or by transforming primary cells with viral oncogenes or chemical treatments. These cells don't follow the normal cell cycle and are able to grow indefinitely. Thus, they are used for long-term research. Growing forms of the cells can also be classified into two. They either grow as monolayers or are free-floating in culture media. For monolayer cultures or adherent cultures, cells are grown in a single layer on a flask or petri dish containing the culture medium. For free-floating or suspension cultures, single cells or small aggregates of cells multiply while suspended in liquid medium.
Now let's proceed to the fun lab simulation from LAB2. Let's go! The first thing to take note of when doing cell culture is aseptic technique, as it is crucial for successful cell culture and for the prevention of contamination. It creates a barrier between the sterile cells that we wish to culture and the environmental microorganisms that may contaminate them. The four key elements to aseptic technique are keeping the work area sterile, good personal hygiene, sterile reagents and media, and sterile handling. In the actual virtual lab simulation, the four steps in cell culture were pre-warming the reagents, seeding a new culture of fibroblasts, performing a cell passage, and freezing a vial of fibroblasts to increase cell stock. In pre-warming the reagents, the first step was to place the DMEM bottle and the serum aliquot to the water bath to pre-warm at 37 degrees Celsius for 30 minutes. Next is to rinse the culture flask and serum tube with 70% ethanol. Then, the culture flask and the serum tube were placed into the hood. Basal culture medium and the serum were pre-warmed in a water bath to avoid thermal shock to cells. DMEM is a common culture media for growing adherent cells. It also has phenol red as pH indicator, which also protects the media from UV light degradation. The serum, on the other hand, provides growth factors to increase cell growth and other hormones. It is usually stored in aliquots inside the freezer for preservation. In the lab environment, we also have the laminar flow hood, which protects the working environment from dust and other airborne particles by maintaining a unidirectional airflow. Despite this, aseptic technique still needs to be practiced. Before putting something inside the hood, it must first be washed with 70% ethanol to kill any contaminants on its surface. And another important rule is to never open any item outside the hood. HEPA filters maintains the clean yet not 100% sterile atmosphere inside the hood. The airflow inside a laminar flow hood is shown in the diagram. The first step in seeding a new culture of fibroblasts was to rinse the culture flask with 70% ethanol and place it inside the hood. Next, using a serological pipette and a pipette controller, discard 50 ml of the medium from the DMEM bottle into the disposal bottle. Next was to add 50 ml of serum into the DMEM culture medium bottle to formulate the complete medium. The complete medium is a resulting medium from combining serum and basal medium. It has everything cells need to survive and proliferate. While the serum provides the growth factors and hormones, the basal medium provides the amino acids, minerals, and vitamins. The complete medium is stored at 4 degrees Celsius for up to 2 weeks to maintain the integrity of its components. Next, using a new serological pipette, add complete medium to the flask. Then discard the serological pipette and put back the pipette controller. Then rinse the cryovial containing fibroblasts with 70% ethanol. Place cryovial in the hood. And using a micropipette, transfer a small amount of fibroblasts from the cryovial into the culture flask. This is known as seeding. Seeding cells also means placing them inside a proper vessel containing the right conditions for growth. The effect of seeding can be observed through the cell growth curve. After seeding, place the flask in the incubator at 37 degrees Celsius. Cell growth in culture has the following 3 phases. The lag phase, where cells are getting used to the new environment after seeding. The log phase, where the cells proliferate exponentially. And finally, the stationary phase, where cell proliferation is greatly reduced or ceases entirely due to space requirements or lack of nutrient. Deviations from the curve indicate an unhealthy environment or a non-functioning component. It is also important to keep detailed cell culture log of the cell growth to detect growth anomalies and to establish the doubling time, which will be discussed in a while. Going back to the effect of seeding, it enables the culture to proceed from the lag phase to the log phase where cells proliferate exponentially. In the log phase, doubling time can also be calculated. Doubling time refers to the time needed for the cells to double their population. It can be calculated by interpolating the growth curve during the log phase as seen in Figure 3. During cell culture, cells are fed frequently to maintain the rate of proliferation. When adherent cells occupy the available substrate and have no room left for expansion, cell proliferation is significantly reduced, which is also known as confluency. Confluence is the ratio of the area occupied by the cells to the total available space. Confluence occurs due to contact inhibition induced by cell surface proteins. To keep the steady cell growth and to stimulate their proliferation, the culture has to be divided and the medium supplied, also known as cell passaging. Adherent cultures should be passaged when they are in the log phase before confluence. Confluence may be detected visually by estimating the percent confluence or the amount of free space, wherein the normal range to perform the cell passage is from 70% to 90%. Going back to our experiment, after removing the flask from the incubator, place the flask on the microscopy stage and observe its cell morphology. The cells observed in the lab simulation were fibroblastic cells, which are bipolar or multipolar and have elongated shapes. The other 2 categories of mammalian cells based on morphology are epithelial-like and lymphoblast-like. The former are polygonal in shape and they grow attached to a substrate in discrete patches, while lymphoblast-like cells are spherical in shape and they grow in suspension. The first step in performing a cell passage is to remove the flask from the microscope and spray it with 70% ethanol. Then place it in the airflow hood. Then empty the flask using a serological pipette. When using adherent cells such as fibroblasts, We need to detach them from the flask before passaging to count them and seed them on a new flask. Common dissociation techniques include shake-off, scraping, and enzymatic dissociation. One example of a dissociation agent under enzymatic dissociation is trypsin, which was used in the experiment. The next step in performing a cell passage is to wash the cells in the cell culture medium with PBS buffer. Then discard the PBS and the medium residue from the flask using pipette controller. Then add 5 ml trypsin to the flask to detach the cells and incubate for 2 to 5 minutes only, since trypsin can be toxic to cells after that period of time. Trypsin is used to break the focal adhesions that anchor the cell culture dish and the cadherin-mediated cell-cell adhesion. Its action mechanism is inhibited by serum proteins, calcium, and magnesium. Next is to add the complete medium into the flask. This is done to inhibit the activity of trypsin, since complete medium contains serum proteins, calcium, and magnesium. Harvest the cells from the flask using a serological pipette and transfer into an empty tube. And finally, add trypan blue and detached cells into a clean microcentrifuge tube to complete the final cell mix. Then add the final cell mix to the counter slide. Then insert the counter slide into the automated cell counter. Then observe the cells in the cell counter screen, which will show that they look different from when they are attached. This is because cells are round in suspension. From the simulations, the cells look healthy, implying the proper observance of the aseptic technique, preparation of reagents, and performance of cell passage. To calculate the cell viability of the cell culture, trypan blue is a vital stain that is permeable to cells according to their membrane integrity. Cells visualized in blue under the microscope will be assumed dead, while the other would be considered as living cells. To utilize trypan blue, add it to the cell solution in a 1:2 ratio. Then, count the total number of living cells and the total number of the dead cells. And finally, calculate the percentage.
In this simulation, freezing our cultured cells required us to resuspend our cells in 9 ml complete medium. We then add 1 ml of 10% dimethyl sulfoxide as DMSO reduces the freezing point of the medium, greatly reducing the risk of ice crystal formation, which can further damage the cells that can lead to cell death. Finally, we can now transfer our freezing medium of 10% DMSO complete medium in a cryovial, which was then slowly frozen overnight in order to store them at -20 degrees Celsius liquid nitrogen.
Cell lines are prone to genetic drift, senescence, and are susceptible to microbial contamination. Since an established cell line is a valuable resource and its replacement is expensive and time-consuming, it is vital that they are preserved for long-term storage. The best method for cryopreserving cultured cells is storing them in liquid nitrogen in complete medium in the presence of a cryoprotective agent such as dimethyl sulfoxide.
We shall now go on to the different cell lines from the American Type Culture Collection. ATCC collects, stores, and distributes standard reference microorganisms, cell lines, and other materials for research and development. The table that is shown on the screen summarizes the different cell lines that were assigned to each group in our class. We will be focusing, however, on the CRL1660 as indicated by the blue arrow pointing on its column. This cell line was the one assigned to our group. You can also pause this video in order to get a grasp on the table, but we will be moving forward to the discussion of the CRL1660 on the next few slides. We call this cell line the Aedes albopictus clone C636 or C636 for short. It originated from Aedes albopictus, also known for its common name, Asian tiger mosquito. Aside from its application for 3D cell culture, It is specifically used to clone flaviviruses and dengue viruses to high titers. It is also a suitable transfection host. As we can see from the picture on the left, the morphology of this mammalian cell line is epithelial, due to its shape, which is polygonal with more regular dimensions. The tissue type for this cell is larva. This cell line is adherent because adhesion is critical in triggering events such as survival, growth, division, and differentiation. To cultivate this cell line, it is required to use a complete medium that contains Eagle's minimum essential medium added with fetal bovine serum. A sub-cultivation ratio of 1:4 to 1:10 is recommended. This is to ensure that in one flask of cells, it will take about 4 to 10 days before they reach their 80% confluency, and when they are subcultured, they can produce up to 4 to 10 flasks of cells, and these subcultured cells can ultimately reach 80% after 4 to 10 days of their subculturing. Split ratios must be consistent for a single cell line, and doubling times of the cell must not be erratic to avoid indication of contamination, cellular death, and wrong choice of medium. Now, let's have a recap on what we have learned today through our objectives checklist. We have defined cell culture as process when cells, may it be an animal or a plant cell, are grown in controlled conditions outside of its native environment. We also know that there are two types of cell culture, and these are primary cell line and the established cell line. We have described the workings in a cell culture lab through the simulation that is provided by LAB2. Remember the emphasis on aseptic technique, as this is one of the major factors that can guarantee us the success of our cell culture. And finally, we went through a run-through of the 3 stages when performing cell culture, which are the reviving of frozen cells by quickly thawing to avoid thermal shock, passaging cells before they reach confluency, and cryo-freezing them in order for us to use them in the future. The 3 major steps are essential as it helps us study different diseases through studying and manipulating such cells. These are our references for the oral reporting. Thank you for listening to our presentation, and if you have any questions for us, just type them on the box and we'd be more than happy to answer them.